Hello students, welcome back and we are going to do our part 2 series on chapter 1. This chapter is a bit many things you need to remember. That is why I split the video into two so that the student can take a break. If you always read the book and you think a lot to remember, it's okay. After you read 10 minutes, you rest for a while, then you come back in 10 minutes, you rest for a while. There's no use to like read, but your body already cannot absorb. Algae, let's look about algae. Okay, algae is has different size. They are macroscopic and micro. Really learn micro. Micro means very small. Macro means big. I mean like in a sense it's big. Big a bit. Macro, is there any word like macro? Macro. I cannot think of the word macro. Okay, so algae, the green color thing, I think you can see this green color algae in the aquarium. Sometimes you see in the aquarium become green, then we say, oh, the algae has grown in the aquarium because there is nutrient, there's light, there's water, so the algae will grow. Sea algae, which can be seen by naked eye, so you can see this is the big algae, macroscopic algae. And there is also a very small one, microscopic algae, which is like Spirogyra and Chlamydomonas. Okay, this one also you need to remember the spelling. Might come out is objective, might come out in subjective. For example, objective, they will ask which of these is algae. So they will give you Spirogyra, COVID-19, and then maybe Lactobacillus. Which one is algae? So you need to circle B, Spirogyra, because this is algae. Basic shape and structure, again, we have unicellular, one cell, or multicellular, many cell. Unicellular, Chlamydomonas. I think this one is easy, but the spelling is difficult to remember. If you can remember, sometimes it's subjective they ask the student write, but it's spelled wrongly, so we're going to have the mark. Spirogyra, okay, Gyra, okay, Spirogyra. Two words is it? Spirogyra. Okay, so these are the shape. Nutrient, most algae are green. Why they are green? Because they have chlorophyll. If have chlorophyll means what? Have chlorophyll means they can carry out photosynthesis by their own. Okay, so here is a brain teaser. Why are algae not found at the bottom at the sea? Yes, very good. Okay, why it's not found? Because here they say algae got chlorophyll, so algae use photosynthesis. So why not in the bottom? Because there are no sunlight at the bottom of the ocean. Okay, so there are no sunlight. So most animals also won't go to the what we call it, the word that means the deep part of the ocean, because there is no sunlight that can enter, and no fish also want to enter because there is no food. Since there is no food, usually animal won't go to there because there is no food. Same like many things are same like bird, or same like bird or lion. You don't see lion live at desert, or bird that fly too high. Usually birds won't fly too high. They can, but they won't, because if you fly too high, what you want to do at there? There's nothing to do, because usually you fly to get to certain place, or you want to get food. So usually bird also don't fly too up high, even if they can. Hybrid algae are fresh, seawater, moist, so again the same thing. Usually all animal or things, living things, like you need water. You need water, you need air, and you don't want but this one is exposed to sunlight. Just now we learned one is fungi no want sunlight, algae want sunlight. This is K-bar question. Okay, so of course if the question asks you why algae need want sunlight, then we explain, okay, because they need to carry out photosynthesis, then of course they want sunlight. Method of reproduction. So we already learned what? We already learned budding. We already learned spore. Okay, we also already learned conjugation. Okay, spore budding is asexual and conjugation is sexual. Okay, sexually. Okay, that is for fungi. And now we look at algae. Is asexually and sexually also is the same thing, asexual. That's why I say if you can write your own notes, draw is a table, it's easier to understand. If cannot, then you think of your own way lah, huh? You draw picture, okay? So bisexually is the same thing, conjugation, but for asexually they are not budding. So I put here fungi. This is algae. For this one, they are using binary fission. So of course, if you got learn computer, you know what is binary. What do you mean by binary? Okay, so this one is I just Google it and we can see that what is binary is a smaller binary digit, a bit smaller unit data, each bit has a single value of 0 or 1. Just now your answer is better, okay? It's at base 2. So you can see 0, 1, 1, 1, 0, 0, 0, 1, 1, 1, 1, 0, 0, 0, 0, okay? So this is a binary, 1 and 2, 1 and 0 only. So it means also that usually binary means only it can, base 2 means it only can develop until 2. So we can see that this is 1 or sexual reproduction. Asexual and sexual reproduction. Okay, so sexual reproduction, they are using, or essentially they are using binary, sorry. Asexual, they are binary. So 1 will become 2. 2 will become 4. 4 will become 8. 8 will become 16. So they are just split. 1 become 2, 2 become 4 and split and continue to split. Sexual reproduction is you can see sexual means you need male and female, but we don't really say it's male and female, but they're going to combine. So 1 and 2 plus 加起来, so they continue to stick together, form a zygote. After form zygote, they will undergo meiosis and then they make many new cells. Okay, so it starts from here, then it will move. This one start from here, 1. Okay, undergo mitosis. We are going to learn mitosis and meiosis. I already learned, is it? We learned at Form 4. So if you still remember, mitosis and meiosis. Okay, so of course meiosis is better because you can create variation. If you still remember, mitosis is you cannot create much variation, so it will be the same as mother face like that means like that, and more prone to disease. This one is more difficult for disease to spread. Okay, moving on. So we already learn a few things. We already learn budding, we already learn conjugation, we already learn binary fission, all these things. So let's look at protozoa. Protozoa is unicellular, means only one cell. Okay, example are like amoeba and paramecium. Okay, so these are the example. Nutrient of protozoa. Okay, Urina carry out photosynthesis, and amoeba obtain nutrient through phagocytosis. Plasmodium is parasite protozoa. Okay, so there are 3 types of protozoa nutrient. It depends. Okay, if you have chlorophyll, then you can carry out photosynthesis, so you need sunlight. Plasmodium is a parasite. Parasite means you eat other people. Amoeba is phagocytosis. He eat by phagocytosis. How to do phagocytosis? This is the example. Okay, first it will make the hand come out. Okay, we call it the pseudopia. To make the hand come out, and then they will use the word engulf. Engulf, the English word to eat the thing, to engulf, to consume. We don't say eat lah, the bacteria eat the thing, okay? We say that the amoeba engulf the food. Engulf doesn't mean only eat, but to like put the thing inside your body, to engulf the food into the body. Then there will be a full vacuum, then the food will be digested. So a few things you need to know is phagocytosis. Then you need to describe the phagocytosis and also the name for the amoeba pseudopia. These are the few things that you need to remember. Okay, so again, these are the example of binary fission by asexual. So 1 become 2, 2 become 4. Okay, sorry, this is binary fission. Okay, so it's asexual. This is asexual, and this one is what? This one is conjugation. So this is bisexual. So you can see there are 4 gamete form that we already learned in the past. Okay, 4 gametes were formed. Okay, so 2 combine together, stick together. You can see here there is a crossing over variation. 这边过去，这边过来. So after that, it will create 4 daughter cells. So these are the okay, so already learn budding, conjugation, binary fission, spore formation. Anything I didn't list out. So basically we already learned 4 types of reproduction. Bacteria is unicellular, again very small. Example, spherical coccus. Yeah, this is the difficult part. This one is a difficult part because you need to memorize this. Because one day I see this question, then I'm like, huh? Because it's very difficult to write the spelling in the answer sheet. So Streptococcus, Treponema, Plasmodium, Bacillus asparagus, Vibrio cholera. These are the example of the name of the bacteria, and you also need to remember the shape: coccus, spirillum, bacillus, and vibrio. So I don't know what the example. For example, the question might ask like, they will write the word, these are the example of bacteria. So they give you a picture. Then the question will ask, what shape is this bacteria? A. Coccus, B. Spirillum, C. Bacillus, D. Vibrio. So you need to circle A. The shape is coccus. So you need to know the shape and the name as well, which is not easy. Okay, not easy. Okay, basic structure of the wall. Of course, they got cell wall. This one you know already. Got cell wall, the shape. Got amino acid, means it's protein. The cell wall is made by protein, of course. Every cell of our body also is made of protein. And polysaccharide, okay, sugar. Of course, it's sugar lah. That's why you also got your cell by eating sugar. I mean, you won't say you eat sugar, you eat rice, but the rice got a lot of sugar. You eat bread, bread got a lot of sugar also. Okay, so we call it polysaccharide. So you need protein and food, sugar, to make your body cell. Some bacteria have capsule which protect the cell wall, known as, how to read this, pili or pili or pili. You know how to read this, pili, which able the bacteria to attach the surface as a shape of bacteria. So this one you need to know. Okay, to ask you to label, you need to label the word flagellum. Okay, why the bacteria need flagellum? So if the question asks you what is the function of flagellum, is to help the movement. It's like a tail but the thing can move. Depend like maybe fungi cannot move, right, because it's stick like a plant, but for this it can move. Got pendulum, amoeba, it can move. Nutrient, again it got chlorophyll. If you take photosynthesis, some are parasite, some are saprophyte, eating from dead organism. Okay, binary fission, this one we already learned. 1 become 2, 2 become 4, and conjugation, this one also we already learned. It's the same thing. Usually they won't ask you to explain. I haven't seen any question that asks you to explain conjugation. Maybe I'm not sure, but I seldom see it. Okay, plasmid resemble a small circle chromosome that replicate independently and is responsible for transforming genetic material during conjugation in bacteria. So where's the plasmid? So in the conjugation, you can see the plasmid. Okay, the small round thing. So what is a plasmid? Okay, plasmid is a small round thing that replicate independently for transforming genetic bacteria. Okay, so of course, like sexual production, you need to transfer the genetic in terms of DNA. So when the daddy and mother married already, of course the mommy and daddy DNA will exchange. Some will give, mother will give a bit to the gamete, some daddy will give the DNA to the gamete. So come out, produce a baby like you. So do you have the same face like your father and mother? No, because you only have half of the genetic material from mommy and half genetic bacteria from daddy. So of course there is some exchange of genetic material, and you are special. In this world there is no second person like you. You are just only you in this world. It's impossible to find the same DNA as you. Wow, very special. Now I only know we are very special. I mean, yeah, you are also very special. Special characteristic as Bacillus arthritis, withstand extreme and very hot place. So some can even extreme and hot place. Usually we say it's water, but this one can grow even at hot place. Okay, so because of why? Because it have protective coat, will rupture the surrounding environment, become a favorable. This allow germinate a form of new bacteria. Okay, so they got like a protective coat, can protect them. Okay, after that, last one. Is it last one? Virus the last one already. I think no more already. Okay, so last one will be the virus. So we already learned about fungi. We already learned about bacteria. We already learned about protozoa. We already learned about algae. Okay. So the second last will be the bacteria, and the last one usually is virus, which we suffer a lot from the coronavirus. But we are not saying that virus is more deadly, because bacteria also can kill people and bacteria also can spread, but just because maybe some virus is stronger, or but bacteria also is very dangerous. Bacteria also, it depends on what type of bacteria. But coronavirus is special because it is very famous for it's easy to spread. Bacteria can deadly, if you got means the chances you die is very fast, but very difficult to spread. COVID-19 you then the next person got the COVID already. So it's very easy to spread. Okay, so the size is the smallest. Virus among all is the smallest, is 0.5 um. So we can see from here, the book say what? 0.5 What does it mean by 0.5 Can I make this thing smaller? It's blocking me. Like this. Okay, then all people can see. Okay, so 0.5 it means that U means 10 times, here is the U, 10 times negative 6. So 0.5 for the virus, the size is 0.5 times 10 power of 6. 10 power of negative 6 equals to the size meter. So this is supposed to be m meter. So negative 6, 0.5, 1, 2, 3, 4, 5, 6. So the dot will be at here. Hey, what am I doing? Cancel. 0.5, 1, 2, 3, 4, 5, 6. So the dot will be at here. So it's 0.000005. This one cancel the dot lah, erase the dot. Okay, so the size of the virus is, if you want to calculate, is 0.000005 meter. Means it is very small. We call it as a micro, 0.4 micrometer. Nanometer is n times 10 power of negative 9, or picometer. The shape is what? The shape, this one you need to remember, spherical, helix, polyhedral, complex. Example, influenza virus, mosaic virus. This one I think no need to remember lah. The name of the virus you need to remember. If you just ask for example, you just can say coronavirus, COVID-19 virus also can. I think it's called the coronavirus, example of the name of the virus. Is there any other name that we can remember, just in case the exam they ask? Don't have, then just put the coronavirus if the exam asks. Okay, so basic structure of nucleic acid thread, DNA, double nucleic acid DNA, or ribonucleic sRNA are protected by a protein layer. So what they are talking about, layer known as capsid, determine the shape and the virus. So viruses are made up from DNA. So you know actually our DNA is very small, but virus is also very small. It's also made up from our DNA. So not only the virus made up from DNA, it start to attack the structure of the DNA. So let's say this is your cell. Why is it so difficult to kill virus? Okay, let's say this is the cell, your body cell, I don't know. The virus will stick on your cell. After they stick on the cell, they will put the genetic material. Okay, they put the genetic material, they change the structure. You know your DNA is only give you skin, but they change your DNA for the cell. So the DNA becomes short already. They will start to change, and the cell will grow. After they grow, they will come out and they will attack your other cell. So it will spread very fast. Attack the cell, spread, attack the cell, spread, attack the cell, and spread. So that is why eventually the person can die, because they are changing the structure of the DNA in your body. So it's very dangerous, like cancer. Cancer also the same thing. You know why we die because of cancer? Cancer also due to one of the main reason is the genetic. Your body genetic material, for example, in your brain, the cell short already. He cannot stop grow, for example. Then the DNA asks him to grow. Then grow. Then inside the brain got lao. Then how do you want to do operation? Is in the brain. So sometimes the doctor open and cut, the person die. So it's a cancer cell, because it is a cell that short already, grow and grow. Usually the cell won't simply grow, but our cell just simply grow, and because it's our own cell, the body didn't go and attack it. So that is cancer. So virus also the same, because it start to change your DNA. So when it change your DNA, it will go and start to attack other people's cell, attack the person's cell, and then eventually the person will die. Okay, virus produced by the cell of their host, and they continue to reproduce. So how to solve this problem? Of course, here didn't say how to kill the virus. Here didn't say. Okay, so of course we have antibody, antibiotic, and we also have antiviral to kill the virus. Now we still haven't developed the antiviral for COVID-19. So there's no medicine, still no medicine for COVID-19 virus. But we wait, I think maybe I think 1-2 years lah. Maybe after a few years, usually they will have hopefully they have the medicine. Then it will stop the disease. That's why one of the way is you want to put vaccine. So there is an early intervention of vaccine to reduce the bacteria of attacking the body. So these are virus. But of course, now it's very high tech already. Okay, they can even create virus to change the DNA so that from bad become good. For example, like cancer is a bad cell. So if you can design a virus that can kill only the cancer cell, so the person will continue to live. So this is called biotechnological improvement. So in the future, they are still doing this thing. They are creating the virus that can attack the cancer cell, but not attack the body. Then we can even find the medicine for cancer in terms of genetic engineering. Now already got already, but now they are still improving it, and also it's very expensive. Yes, correct, very good. Okay, so of course, during Corona pandemic, you want to boil the water so that it will kill the bacteria, kill the microorganism. For example, like Vibrio cholera to clean it lah. So this is a corona epidemic, means it will spread usually to the stomach. Okay, the stomach feel pain, then you will have the disease, fever, lao sai, and so on. So of course it's very important to boil the water. But nowadays got many high-tech water filter, so usually we don't really need to boil the water. But if you want, you can still boil the water. Okay, so this is the experiment. So we bear with me, maybe this is the last one. So after we talk about the experiment, then we will conclude today's topic. So sometimes in the exam, they will ask you to discuss the experiment. For example, they can ask you many things. So I need to teach first. After I teach only, I give some example of question they will ask. Okay. So let's say we want to do experiment. What experiment that we want to do? What you want to do? So let's say we want to do the experiment is to compare the growth of bacteria in sterilized algae. Wash hand, wash with water, and wash with water. So now one day one student asked, because it's a science class, they asked, teacher, you say that the water use soap is better than water, how you know? Then the teacher say, oh, you need to do experiment. So the student asked how to do the experiment. Okay, so in order to do the experiment, first we need to have a hypothesis. So what do you think? I asked the student, what do you think? The student say, if I use soap, I think the bacteria is lesser to those water that I do not use soap. So it's a relationship. Usually it's a relationship. But assume, you don't know one, you just agak-agak only. So you agak-agak, when the clean of water finger is streaked with the surface of sterilized algae increase, bacteria grow on the surface of the sterilized algae will decrease. What is this? Cleanliness level of finger streak the surface of the sterilized algae increase. Bacteria grow on the surface of the sterilized nutrient will decrease. What is this? Let me read again. So long one. When the cleanliness level of the finger restrict the surface of the sterile nutrient agar increase, the bacteria grow on the surface of the sterile agar will the bacteria grow on the surface of the okay. So okay, for the experiment you need to understand the MV and the RV. Okay, manipulator, and I just make it simple lah. I make the hypothesis simple because I read a few times. I maybe I change the word a bit. Okay, so the hypothesis I say, when the cleanliness of the finger with straight the sterile agar increase, when the cleanliness of the finger with straight the sterile nutrient agar increase, the number colony of the sterile agar will decrease. Correct or not? Means if your hand is more clean, then the bacteria is lesser. So this is the example of the hypothesis. It's a relationship. When the finger is more clean, restrict the sterile agar, the number of bacteria colony will decrease. That's why when they write in the sentence, you see when the cleanliness of the finger straight the surface increase, then the bacteria grow of the surface agar will decrease. Okay. Oh, that is why it's saying that if your hand is more clean, less bacteria. If your hand is more dirty, then it is more bacteria. So they just copy A and B to the hypothesis, but when you read it makes confused. Maybe he should say when the cleanliness of the finger that touch or strict the sterile agar increase, the number of bacteria colony will decrease. When the restrict the surface. Okay. Okay, so it's saying the same thing lah. Yeah, okay. So this is very important. In experiment, you must always understand hypothesis and variable. If you cannot understand hypothesis and variable, you cannot do the question already. You straight away will get 0 mark. Because you don't know what you are doing, what you want to do in the experiment. So if you don't get your hypothesis correct, then you cannot do your experiment, because at the end of the experiment is your assumption we want to prove. 你要证明这个东西 want to prove, because we are doing science. We are not doing like a religion or philosophy that what do you think or what do you think. It can be right, it can be wrong. But when we are talking about science, we cannot say that we must do experiment to prove it. If you cannot prove it, then you don't put in your textbook, because it's not science. That's why you see science, they seldom talk about religion, they seldom talk about ghosts, they seldom talk about philosophy. I mean they talk, but in a more concrete strength, means you must prove it. So now they make a relationship, say okay, more clean means less bacteria, constant is the temperature to make the experiment fair. Material, okay, put a sterile nutrient agar. They put the word sterile means it must be clean, means already heat it, it is clean already. You cannot use non-sterile nutrient agar. If you use just simply nutrient agar, then all bacteria also will grow, because it is not clean. So you want to make it clean, you want to sterilize it, maybe you just heat it with fire first, or make it hot. Okay, so, and cellophane tape and marker. So label it A, B, C, D. So carry out strict the entire nutrient agar bacteria with unwashed finger. So A is 没有洗手 unwashed hand. Figure B, wash your hand with water. So this one is wash hand. Wash hand and touch the B. And C is use the soap. So C is you use the soap. And D, I think is the control variable. D, okay, so D is why he didn't touch. D is a control. Right or not? Control. Means you don't want to touch it. It's a control experiment that you do here. You want to control it. The temperature control. Because why you want to put control? Let's say here grow bacteria. Here got bacteria, means this whole experiment failed already. Cannot use already. Because it is a control. Then why control you got? People will ask, why your control variable got bacteria? Means something wrong with your experiment already. So we need to do again and test again. So of course in science we always have a control variable. We just want to put it there, we just leave it there, see got problem or not. So if the control bacteria no problem, then we watch here, okay, here more bacteria, okay, bacteria, no bacteria, then we conclude the experiment. Or maybe here more bacteria, here more bacteria, here no bacteria, also can. So we can say that unwashed hand and washed hand also got bacteria, soap no bacteria. Or here got bacteria, got bacteria, got bacteria. So it also means if 3 of these has bacteria, it means the hypothesis need to reject, means it's wrong. It means that if the hand is clean, it does not relate to the growth of bacteria. So you need to do experiment. Okay, but of course we in our heart we know that usually here is more bacteria, okay bacteria, less bacteria, no bacteria. So it proves the hypothesis is correct after we do the experiment. Okay, so this is a control variable, and then we have the cover petri dish and put a cellophane tape. You want to put a cellophane tape? Oh yeah. So after they close it, seal it with the cellophane tape. Okay, invert the petri dish. So after they close it, after you touch one time, close it, put cellophane tape, wash the hand, touch it, close it, put the cellophane tape, because you don't want the bacteria to go in. Then people will ask you, why you do the experiment, you fail already, why you didn't, how you know the bacteria didn't go in? Oh, because I put cellophane tape to tape it. And then after that, you go and use the soap, wash the hand, put it, tape it. So people cannot ask you, is there any external variable like the presence of bacteria that affect your experiment, means your experiment will fail. A, B, C, D, just write. You want to put 3 days, you want to put 5 days, you want to put 1 week, you want to put 2 weeks, up to you, okay? Doesn't really matter, you still get correct as long as you can say it properly, but you don't say 1 hour lah. 1 hour the bacteria also haven't grown, okay? So you use your own word lah. When the exam asks you, you can just say you want to put how many days, up to you. After 3 days, A, B, C, D cardboard, then you need to do your observation, tabulation of data. Here got answer or not, but we can roughly guess lah, okay? Number of bacterial colony, maybe we put here 4 bacterial colony, here we put 3 bacterial colony, here we put 2, here we put 0, up to you. Or you want to put here 5, you want to put here 4, you want to put here 1, you want to put 0, you want to put here 10, you want to put here 5, you want to put here 2, you want to put here, up to you. Okay, actually you no need to put, but if they ask you to assume, then you just write, okay? Because sometimes they just give here 10, here 3, here 0. So what is your answer? So you can put maybe 9 to 9, 8, 7, 6, 5, 4. You don't put 3 lah, or you don't put 10, okay? You just agak-agak you put your answer. So after you do the experiment, now the student and the teacher can explain. The student can tell, oh teacher, you are correct. After I do this experiment, teacher, I found out that if I didn't wash my hand, the bacteria is growing more. If I wash with water, the bacteria is lesser. And if I use soap, it will be even lesser. Then the teacher say, yeah, very good. Now we learn that not only we do the experiment, we prove that we prove what? If the hand is more clean, then the bacteria will be lesser. So this is a science experiment. So we already proved it. Then we got the conclusion. So after the conclusion, what will happen? After the conclusion, we will put in the textbook. Then you can read the textbook. Oh, like this. So whenever you take a textbook, actually it consists 10,000 of experiments that other people do. What is this picture? Okay, 10,000 of experiment people do that put in your textbook. Now we are reading one of the textbook. Actually already thousands of experiment people do. They don't simply put it in the science textbook. They already do the research. They already do the experiment. They do the discussion. And then they then they put in your textbook. Okay. Of course, here he didn't put the discussion. When you go to university, there's one part for here before the conclusion there is discussion. Then you need to explain why. Or because it grow because of the bacteria name is blah, 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 what day, what time, and this grow a lot because of why it grow. Very long discussion. Okay. Then from there you read, then you understand. Then we have the conclusion. Okay. So basically that's it. Okay, so if no question, I teach until here only, okay?
Okay.
Okay, so I see you again next week, 8 o'clock. See you again. Thank you and bye-bye.
Thank you, teacher.
Bye-bye.